everyone. My name is Elizabeth Abidi. I am an honors biology student, part of the Truman Lab, and today I will be presenting my project on the role of HSP70 phosphorylation on cell integrity signaling in yeast. And so for the talk layout, I will be discussing my background information on HSP70 and the cell wall integrity pathway, the current data I've collected thus far, the future experiments and implications of my work, and the acknowledgements of my lab members and others. So what is HSP70? Well, HSP70 is an important molecular chaperone. It's also known as the heat shock protein 70. So it is responsible for folding newly synthesized client proteins. And here we have a schematic of what exactly its mechanism is. So we have an unfolded protein that gets unfolded due to some sort of proteomic cellular stress. And it will basically be signaled by the HSP40 co-chaperone molecule, and it will bring it to the HSP70 to ultimately result in a newly folded active protein. And this protein is highly conserved amongst all organisms, so it's seen in mammals and microorganisms as well. And in order to complete its necessary job and function, it requires the assistance of other co-chaperone proteins that I mentioned before, such as the HSP40 and the HSP90. And the HSP70 is also known as SSA1 in budding yeast. So I will be using those terms interchangeably, but they essentially represent the same sort of molecule. And HSP70 is also regulates many signaling protein pathways in yeast, and one of them include the MPK1 kinase, and that's part of the cell integrity pathway. So what is the cell wall integrity pathway? Well, it's a MAP kinase pathway in budding yeast that helps maintain the structural integrity of the cell wall. So here, when the cell wall is under some sort of attack, usually it's due to a stress such as heat or caffeine. It causes the cell wall integrity pathway to act like as a security system, and it will cause a series of phosphorylated kinase activity intracellularly, and this will ultimately regulate the amount of cell wall specific genes responsible that will compensate for the external stress and other environmental challenges that the cell had to face. And here you can see MPK1 is the last kinase responsible in that phosphorylation process, making it a very important molecule to study. And so what's unique about HSP70 is that it is modified by post-translational modifications. So often we hear during protein synthesis, the central dogma theory, how from DNA it turns into RNA, from RNA we get a protein. However, post-translational modifications is an important step that's often overlooked in this process, but it can have very intimate alter function of the protein. And so here we have modifications on HSP70, and there are over 80 PTMs that are seen on its structure. And one of the most highly regulated one is the one highlighted in pink over here, and that's phosphorylation. And however, a lot of its regulation and is not really understood or well known in scientific studies. So we're out here to seek what its role could potentially be in cell signaling pathways. And so that leads for the project aim to explore how those HSP70 modifications could alter and fine-tune the activity of the cell wall integrity pathway, but specifically the MPK1 kinase pathway that gets highly phosphorylated at the very end of it. And so here we use yeast as a model organisms to study chaperones. So often we know yeast that's involved in the bread we eat or the beer or wine we drink. However, yeast looks very similar to what you see here. That's in a lab setting. It looks significantly different. But what's unique about this sort of model organism, it's very cheap to grow and easy to genetically manipulate and culture. It grows and divides extremely fast. And the human and yeast proteins are highly similar, making it a great candidate for us to use within the lab. And so what we have done is we wanted to screen for this chaperone co-collection for caffeine resistance. So we have taken each specific phosphorylation on SSA1, and we had either mimicked or permanently modified the phosphorylation states. So for the 73 phosphorylation sites, we mutated them to either an alanine, which is a nonpolar amino acid that can't be phosphorylated, or an aspartate or glutamate amino acid that can mimic that phosphorylation state. And then we screened all 146 mutations for growth at standard temperature 30 degrees Celsius and at different levels and concentrations of caffeine, specifically 5, 10, 15, and 20 millimolar caffeine. And we plated them on Petri dishes so we can observe their phenotypic response. And so here we have a result of the serial dilutions of HSP70. So we screen them for sensitivity to specifically caffeine. And here you can see this is our control at 30 degrees Celsius. And our experimentals over here, 5, 10, 15, and 20. And these sites you see highlighted in blue are potential ones that could be responsible since they are significantly sensitive to heat. You can see as the concentration levels increase, we have a decrease in the phenotypic response. So there's some sort of defect on the amount of HSP70 expression. And so these 10 sites were sensitive to the caffeine. And we wanted to further conduct an additional analysis to better understand the physiological significance. So we have mapped the sites onto the structure of yeast HSP70. And so just before I do that, I would like to give a little background information on the architecture of the protein. So here in orange, we have the nucleotide binding domain that is responsible for the hydrolysis of ATP. And then it transmits that signal through the linker region and it rearranges it, allowing the SBD to basically unfold and bind to the misfolded proteins within this hydrophobic pocket over here. And the C-terminal domain highlighted in blue will basically clamp over that region, securing the substrate, allowing it to complete its process. And so the results showed very promising. And actually, we has opened up a lot of doors and questions about what exactly these mutants play a role on, because a lot of the mutations were seen on the MBD terminus of HSP70, and we actually have seen them on the peripheral as well. So potentially these sites could play a role in possibly the ATP hydrolysis aspect of the protein, and that could potentially alter the signaling as it passes through the protein, which could potentially impact the way misfolded proteins can bind to the client region over here. However, we did see a few interesting ones as well. We saw one on the linker region right here and on that substrate binding domain. And so we're not exactly sure what this could essentially mean, or what is the reasoning behind their specific location. However, we would like to move forward. That was something we would like to look and explore a little more of. But so far we have demonstrated novel insight on the importance of the HSP70 phosphorylation in yeast stress resistance. And we have shown that by demonstrating those 10 sites that were very sensitive to caffeine. And since caffeine is a well-known stress for the cell wall integrity, this could potentially show a new role for HSP70, acting as a major integrator for phosphorylated dependent pathways in cell signaling. And however, we're not 100% guaranteed that these sites may directly being impacting the cell wall integrity pathway. This might provide further evidence for possible crosstalk cell signaling, so multiple other MBK or MAPK kinase pathways could potentially be playing a role in crosstalk. So we could see other pathways basically intertwining and impacting the same sort of sites or the same sort of pathway. And then so the next steps, we would like to explore a little further about how this occurs downstream. So explore further the mechanism associated with it. So we want to conduct a series of immunoblotting techniques to detect phosphorylation expression of MPK1, and then we would like to subject those specific residues and sites to transcriptional analysis to really quantify the amount of RLM1 transcriptional levels that are associated with the phosphorylation sites. So ultimately, this project could be significant to understanding the role of external stress on and environmental challenges that our cells have to face. And I would like to ultimately thank my lab team members, my mentor, City Omkar, and my uh, PI, Dr. Andrew Truman, and the rest of my lab members for just being so welcoming and for allowing me to participate in the field of research. Thank you.